Hello there, evolution students at IPAC EDU. It has been a while since I've managed to complete a lecture. Today's lecture is going to be among the most complex and hopefully interesting lectures in the principles of evolution. So far in this class, we have reviewed the early ideas of evolutionary thought. We went into detail of Darwin and Wallace's contributions. We looked at major evolutionary transitions on the tree of life. And we focused then again on the same in animals. We did a genetics review. Then we jumped into population genetics, where we looked at the forces of mutation, migration, drift, selection, population size, and population structure on the patterns and processes of evolution by descent. We went into detail on natural selection. We revisited population genetics again, looking at adaptive landscapes and fitness tunneling. Then we looked at selection at multiple levels, interdemic selection, selection at two loci, and kin selection. I taught you what I know about the evolution of quantitative traits, norms of reaction, phenotypic plasticity, and gene by environment interactions. A little bit about that. Then we went into the neutral theory and nearly neutral theory. Then we went into the neutral theory and nearly neutral theories of evolution. Um, and we had a lecture most recently on the genetic and genomic and protein evolution, the evolution of genetic systems. And this has been a fascinating 14 lecture journey so far. And today, going into the evolution of complex phenotypes and regulatory networks, we're going to game up a little bit, given that you have all that background. If you're new to IPAC EDU, this is a free lecture that's going to give you a taste of the level of detail that we go into teaching complex ideas. And if you don't understand what I'm talking about, you can go all the way back to Bio 1 and Bio 2 as if you're a freshman in college, and you can take those classes. We also have a 2-semester class in genetics, which covers all of genetics. And that's a wonderful class. You can sign up for both of those classes, so all three of them actually, four of them actually, starting this fall. It's 2024, and you could take Bio 1 and then Bio 2 in the spring, and then jump into genetics. I will let students, participants do their courses in a self-paced option if you'd like to just walk through the lecture material on your own. But we do have live class meetings so that you can hear questions from other students and get involved in the discussion yourself, which I think is important. So let's get on with it. Today's lecture, The Evolution of Complex Phenotypes and Regulatory Networks, demands of us that we give a definition of genetic regulatory networks. These are systems of interacting components, genes, proteins, small molecules that govern gene expression levels in a cell or organism. They are key to understanding cellular processes, development, and responses to environmental changes. If you haven't had molecular biology, this lecture will give you an upgrade in that way, embedded, because to understand the cellular processes, such as methylation, cell cycle development, growth, maturity, physiology, metabolism, immunity, tissue identity, tissue function, and organ function from a molecular standpoint, you have to know some of the lexicon. So I'll try to make sure that I define all the terms that I'm using. So given their importance in the organism or the cell's ability to respond to stimuli and maintain homeostasis and control development, I wanted to take this approach where we go back to the beginning of the tree of life and not step through all of the tree of life, but in general, in a very general way, take a look at the phases of evolution, the development of regulatory networks from the simple to the complex. And if you're into complexity theory, you're probably going to find some gotchas on me. I might be making some assumptions or misuses of words in complexity theory, but that's okay, because we'll have courses on complexity theory and nonlinear dynamics and other really highly advanced analytics here at IPAC EDU just for fun. And so, yeah, just to learn. But the process, you'll learn that they involve the first regulations, those that have survived anyway, the first regulatory systems, processes like gene duplication and mutations that happen after gene duplication on both the signal and the receiver parts of the regulatory networks. And then, I don't want to say this allows specialization. You might hear me go into that at points in time, but that is a bit teleological for me. And so what I'd like to say is, as a consequence of these processes, then we see functional specialization of different proteins and refinement and optimization of the expression of the genes that encode them, and long coding RNAs and tRNAs and everything else that's read off of the genome. Um, we're going to talk a little bit about at the end of how crucial it is to understand genetic regulatory networks for biotechnology, medicine, and synthetic biology. And those of you who know me, I'm Dr. James Lyons-Weiler. I eschew modifying heritable variation in humans. I really think that's a bad idea. I don't mind modifying heritable genetic variation in other organisms to try to understand the basic biology for this, for the sake of knowing and asking questions. And certainly would celebrate any safe and effective genetic modification of humans that have, dire illnesses that are absolutely genetic and congenital. My gravest concern is the modification of the heritable genetic variation, because I believe that ironically is the end stage of the genetic regulatory network control. Who's controlling genetic regulatory networks now? Biological optimization, aka natural selection. Should we change heritable variation for intellect and strength and size and any peculiar personality traits that you think that you might be able to decode from the human genome? I think that's a matter of the question. We're standing on the threshold here. There are certainly people who would like to do that, transhumanists and so on. And we've had entire courses on that as well. The perils of coding humans, for instance. But if you're interested in that topic, you can go read my peer-reviewed paper in biological theory, Who Are We and Who or What Do We Want to Become? It's an in-depth look from an evolutionary standpoint, citing evolutionary theory and evolutionary knowledge, and all the greats in evolution. If you're great in evolution, you happen to view this video and I left you out of that paper, I'm sorry. I apologize. Can't cite everyone. We know this, right? But the perils of changing heritable variation are not going to be further addressed in this lecture. So that particular paper is Who Are We or Who or What Do We Want to Become? I published it a couple of years ago. And have a look at it and see what you think. I promised you that we're going to talk about the evolution of complex phenotypes such as the eye and the vertebrate limb, flowering development of plants, maybe something about color patterns in animals and immune systems. We do have an entire course on how the immune system works. It's the biology of the immune system. It was one of the most fascinating courses I had the pleasure of putting together. But I want to warn you that, again, this lecture is not going to be, say, part of the argument that evolution can't occur because something as complex as the eye could never evolve overnight. It's pretty clear that evolutionary biologists understand that the eye per se does not evolve overnight. We have had plenty of time for eyeballs, and the specific eyeballs we carry around in our head to evolve, and they're so valuable that they've basically the same form of eye has evolved more than once on the evolutionary tree of life. If you look at an octopus's eye, it's remarkably similar in form and function. And so, yeah, this course is not really about justifying or rationalizing or trying to convince anybody of evolution. It's really about teaching. And I believe that the public is not as well off as they could be if they don't understand the principles of evolution, but I believe that it's their choice on whether or not they want to understand it. There's no moral judgment on my part. So let's get into some of the complex processes of genes interacting. This is important because at one level of epistemology, the understanding of what's happening at a given level of inquiry really depends on what you can measure. If you're out in the field and you're an ecologist and you're looking at, say, maybe you're even looking at inheritance patterns of phenotypes, you might see that there's correlations between specific, flower shape and color or something like that. But you might not be able to nail down the actual specific mechanism, the molecular mechanism. Certainly, geneticist William Bateson in 1907 couldn't. He didn't have a full understanding of genetic regulatory networks. But epistasis is an important term that involves how genes interact with each other. And, this was observed specifically in butterflies and in plants and inheritance patterns. And, we have a effect of a gene mutation being dependent on the presence or absence of other mutations, so other variation. Um, so, and then, if you try to understand what a gene is versus a mutation, you have a gene that encodes a protein, and that protein may have a particular function, but it will not have that function or will have more or less of that function depending on the presence or absence of another type of genetic variation at another quote unquote gene. And so, gene is, in my view, an inheritable piece of information in the human genome or any genome that has some effect on phenotype ultimately, and it's heritable. So epistasis is something to keep in mind, but it's not at the right level. These ideas like epistasis and pleiotropy, where a single gene can then turn around and affect multiple phenotypic traits, again, it started in the early 20th century, and all the work on Drosophila, the fruit fly, showing that variation at one particular gene might have a necessary or even a potential consequence on the phenotype, how an organism looks. They're done at the organismal level with patterns of inheritances, let's say through pedigree of bred animals or plants. And so understanding this then becomes tenuous in humans because we don't have that degree of control of experimentation, and that's a good thing. So many of our genes may have multiple effects on phenotype, and we don't understand them completely. But there have been some cases where there have been genes that have been studied by different teams because they have different functions, and they were given different names, only later on to find out that those genes actually are the same one and the same gene. And so, yeah, it's certainly we can kind of fall into understanding it. The earliest clarified example of a genetic regulatory network is the lac operon. It was first clarified by Francois Jacob and Jacques Monod at the Pasteur Institute in the 1960s. The lac operon provides a clear model of how one gene can be regulated by another in a coordinated manner. And this is a pretty complex system that involves the lac operon, genes, promoters, operators, and repressors. So the lac operon's primary function allows bacteria to switch the phenotype so that they can actually use lactose efficiently under varying nutritional conditions. So here we have, when glucose is present, a protein called the catabolite activator protein that comes in, activates a promoter. Now that promoter is going to be, sometimes I'll call it a gene regulation region or a regulatory region or a regulatory element. The promoter is usually 5' to a gene, but it doesn't have to be. It can be found in an intron or sometimes even in an exon in a gene. But there's a release of a repressor that then allows the binding of an operator. But when glucose is present, the inducer repressor then is released. Okay. And so you have, when lactose is present, lactose through the CAP um, binding the inducer repressor causes its release. So you see that there's a little bit of lactose there, uh, that little piece of lactose there that actually binds to it. So you end up with um, very little mRNA when glucose is present and lactose is present. But then in the third case, where glucose is absent and lactose is present, then you have the cyclic AMP that comes in and binds to the catabolite activator protein. And even though that little piece is there with the repressor, you end up getting abundant lac mRNA, you get the lac protein, and then you can go ahead and metabolize, get energy from lactose. So you can see that basically what we're looking at is a phenotype that's conditional on the environment, with a promoter region that's affected by another protein from another gene, the catabolite activator protein. There's also the repressor protein and the operator protein. So we're looking at a fairly complex system. I'm not going to ask you to memorize this. This comes from the Biomodels website, way back in 2006. You can go to their website at that link down there, and read all about this. So in a broad view, the components and functions of gene regulatory networks include genes, DNA sequence that code for proteins, transcription factors. These are what I would call regulatory elements, proteins that guide gene expression by binding to specific DNA sequences. And then other regulatory elements, I throw transcription factors in with regulatory elements in the categorization. You have promoters. These are specific sequences on the genome, promoters, enhancers, and silencers, that then go over and bind to, okay, the element. And we saw this here. So we have a repressor and so on, inducers, repressors. And so these GRNs control the developmental processes like cell differentiation and organ formation. They regulate physiologic responses to internal and external stimuli. From an evolutionary standpoint, the early regulation is seen earliest in archaea. So the first regulatory genes that we see here, um, that come from the last common ancestor of all the tree of life, include the TRMB family transcription factors. They regulate sugar metabolism in archaea. Heat shock proteins protect cells from thermal stress. And the basic regulatory mechanisms are, as you've seen, transcriptional regulation to an environmental change. And this gives us evolutionary insights into the evolution of complexity in the big kingdom of life there. So, if we go back to the very early work here in bacterial survival, of course, this is a very good example. And there are others within then our archaea. Maltose, operon B is an example of transcriptional regulation in the archaea. So then, it's 2024, not 1907, and we have a lot of knowledge, and there have been people who are working with GRNs for decades, really, since the molecular revolution in biology, the age of molecular biology. So in addition to genes, transcription factors, microRNAs, these all will work together to form a particular structure, so a network of a structure. And that structure, of course, is a representation of the knowledge of how that network works. And, there's a degree of structural complexity due to the diversity of components. The more we study and the more we choose to understand, regulatory networks can interact, and so then they can become unwieldingly large if we try to spell out the entire regulatory network of some phenotype or some particular aspect of an organ's function. And the main point here is that they really enable the sophisticated control mechanisms for cellular function and adaptability to environmental change. One of the topological features that we see in gene regulatory networks include loops. So there's feedback loops It can help regulate gene expression stability. Negative feedback loops, for instance, maintain gene expression within limits. So there's every time you do a transcript product off of a gene, there's feedback up to a threshold. After that threshold, then there's feedback that says, okay, guys, shut it down. And then there's feed-forward loops. These involve sequences where one gene regulates a second, and both are jointly regulated by a third. I'm going to give you an example of a feed-forward loop if you're not familiar with it. These are important for filtering noise and for creating time delays in gene expression. Timing is very important. Having a gene and expressing it is one thing. Having a gene and expressing it at the right time and creating the right amount of protein. This is the degree of control that we have. And when it goes wrong, we have, pathophysiology of disease. So the feed forward control block diagram here, imagine some input information, YSP. In the presence of feed forward control, you have a product U, it undergoes a process. And then the process, the amount of Y that comes out of that, which is a modified YSP, is actually dependent on that process occurring. And then in order for the second part of the process to keep up with or slow down with the product U, right? The U itself is not the signal to do that per se. Then if U itself doesn't control itself, it's not a self-regulating feedback loop, then the feed forward has a directing element that comes in and upregulates both the feed forward control. Yes, YSP is going to be transformed to U, but then it also has to upregulate the second process so that U can be processed into Y. And this is a good example. These have evolved within genetic regulatory networks. Another topological feature of gene regulatory networks include modules. There are semi-independent clusters of genes. They execute the specific functions. They're controlled by distinct regulatory mechanisms. And modules might interact through their products. But there may not be any feedback and control back between the two. All right, so this is a degree of modularity, and it allows isolation of the control. So there's not the more complex you get, you could enter into a situation where you can become chaotic. Modules ensure robustness, a certain degree of resilience, and maybe even redundancy and flexibility in responding to environmental conditions. So an entire gene regulatory network of genes and proteins can exist in an organism under one environmental condition. Only then that entire network is then shut down and you can switch over to another. And then this is one of those teleological things, but people tend to think about it as, oh, with this kind of modularity, then you can explore the evolutionary landscape. And I want to again avoid teleology and say organisms that have modular gene regulatory networks, therefore, end up exhibiting a higher amount of evolutionary innovation. Right. So there's no anticipation. They don't exist. So therefore, so that they can. They simply do it as a result of having those features, and that's important. So feedback and feed-forward loops. We can look at the lac operon, which we just did. That's definitely the presence of a feed-forward where the lactose presence is a trigger. It activates the promoter and the operator as well. You saw that in the last thing. And then if we think about modularity and evolution, we're going to look at the vertebrate limbs, but I chose this particular piece of artwork to showcase that there's a level of complexity here that we are working very hard to understand, where we want to understand it. Probably by now it's understood best in the brain. I would imagine, with the brain studies that have been done to simulate brain function. But the degree of modularity that we see in complex say, heterozygous organisms have is truly astounding. It really is truly astounding. I mean, people have based their entire career studying one module and how it interacts with others. Um, so looking at an example of vertebrate limb development here, um, part of the modularity allows a conserved core network, and there's that word allows, but part of it has been observed to have a conserved core network. There's a core set of genes and elements that are highly conserved. And in this case, across vertebrates, they're crucial for initiating limb development. And there's a number of genes, including TBX5, FGF10, and SHH. And then rather than having new genes for limb development in vertebrates, so you start with fish in the water, you end up with vertebrates, tetrapods that are walking. You may certainly see protein coding sequences change themselves, but the primary effect here for limb development is how these core genes are regulated and when. And we're going to see this in some example here. So I do give the reference for this at the end, but in this particular image, um, we can compare, say, a chick embryo and the lungfish and Pisces, another fish, um, Ambystoma, amphibia, and Xenopus lavis, amphibia. And we can see that the same genes are involved in fin and in limb development when you actually look at the gene expression. So 400 million years ago, animals moved from water to land. The tetrapods. The tetrapods are a group that includes all four-limbed vertebrates, from frogs to shrews to dinosaurs to humans. And if you want to understand whether the tetrapods are monophyletic, that is, they derive from a single ancestor, or paraphyletic, that is, they evolved multiple times, or even polyphyletic, then don't study the organismal genes for vertebrate limb development, because it's very likely that very similar regulatory pathways will evolve in a convergent manner. Now, overall bioplans include HOX-A and HOX-D clusters, and these turn out to be important for limb development in mammals in particular. Their expression divides it limited to regions: a proximal region closer to the body and distal region, including the digits. But the recent research on Hox genes, and it's been known for quite some time actually, shows that the gene spatial arrangement of chromosomes are essential for the regulation. You start moving them around in sequence, then you get different aberrant development. But the whole point here is that the gene regulatory network, these pathways are repurposed through regulation, not necessarily through proteins that encode, specific proteins for the overall bioplan. For things like, refinements, there may be, certainly plenty of room. There's certainly plenty of room for protein evolution as well. But if you take like a zebrafish and you take their genes and you engineer transgenic mice with genomic regions of zebrafish, the HOX-A and HOX-D genes were consistently expressed in the arm, even if the transgenic region activated genes in the distal part of the fish's fin, which means that they correctly, put the right genes in from the fish into the mouse, and they still got an arm. And so that's regulatory control rather than the specific proteins. And surprisingly, the same spatial regulatory mechanism that controls HOX-A expression was present in the genome of the ancestor of modern fish and land animals. So you can see that the stylopode, the zygopode, and the autopode in the vertebrates, the mammals, have ancestral specific bones that are their corollaries or homologous to. Right. So that retention of homology of specific bones means that the limbs did not evolve de novo out of the blue by protein evolution, gradually improving something out of the blue, but rather refinement on an existing theme. So with the conserved core network and then the regulatory changes that we understand, there's also modular modifications that can occur. So specific changes in the GRN can occur without disrupting the entire network. An example would be the apical ectodermal ridge module that can affect limb outgrowth, while changes to the zone of polarizing activity can influence digit patterning. So the degree of refinement, in a way, are like nested sets of control where you can change part of it, but being not a perfect nested set, then there's a degree of autonomy without feedback among the other modules. I hope that's clear. So then existing developmental modules can be redeployed in new contexts. So a completely new environmental challenge can cause a lineage to do better in that new environment if they have something to work with. And why not redeploy an existing module? Well, that's what we see time and time again, over and over and over again. We don't see the development of brand new modules per se all the time. We certainly do, but we don't see it all the time, like within a particular degree of granularity on the evolutionary tree. So you can change you can see predictable morphological differences based on timing or spatial expression of key genes. So like extended expression of certain genes can result in these elongated digits like we see in bat wings. And then comes, this concept of evolvability, which again, I try to eschew it, but it's in the literature, so I want to be fair and give it a shot. But evolvability is the allowance of the exploration of a greater amount of the phenotypic space, if you will, in the exploration of adaptive niches. And so without modularity, organisms might not be able to respond as well. Do the modules exist so therefore organisms can be more evolvable? I'm not sure the evidence is there for that. But when you run into this, if you do your own reading on this, you'll be able to put that perhaps into context. But the conserved core network can also be seen as a constraint on variation, right? We don't see much variation among limbs. If you look at a bat wing and you look at your hand, you can see similarities. If you look at a whale fin, when a whale being a land animal that returned to the sea, you can see the 5 digits. And so inheritance by descent with modification is done via the constraint provided by or that occurs due to modularity in gene regulatory networks. This is that question again. It raises its ugly head, whether or not this then allows rapid adaptation. It's not really necessarily allowing rapid adaptation, but perhaps this is a way that, rapid diversification could occur if the niches are there to receive the variation by reprogramming modules. And then, as I said earlier, it's possible tetrapods evolved more than once. The phylogenetics might not say so. It certainly is conceptually possible, where the modular changes can lead to convergent evolution because the same events become very likely to occur, or very phenotypically very similar events might appear to be very likely to occur in distantly related species facing similar environmental pressures. Binocular vision in predators is one, right? There's plenty of examples of that. And then this idea of evolutionary tinkering, where it's a tinkerer rather than an engineer. It puts forward these variants upon which natural selection works, and the optimal, not the perfected. It's not about perfection, but through competition within evolving lineages within a species, we can see some surviving better than others and reproducing more than others, or reproducing less if that's the the optimization function. I did not, I've talked about it a lot more than I expected that I would, but the evolvability here might have a good explanation in terms of what researchers call buffered qualitative stability. It's a theoretical framework developed to explain how gene regulatory networks maintain their robustness and evolvability. So you have to assume evolvability means here that lineages are more likely or not more likely to diversify given evolutionary pressures and challenges and opportunities. But the buffered qualitative stability idea posits that the organization of gene regulatory networks allows them, there's the allow, to remain stable, that's resilience, despite environmental and evolutionary changes. This stability is achieved by ensuring that the network's qualitative responses are maintained, even when quantitative parameters such as protein concentrations fluctuate. What the heck does that mean? Well, the qualitative stability is the ability of the network to maintain stable gene expression patterns in response to certain perturbations. That sounds like a good strength to me, right? Buffered stability, the network's resilience to structural changes, such as the addition of new regulatory links, means that it can take on new functions by adding pieces without necessarily upsetting the entire apple cart. Right there is where I have a problem with evolutionists that think that evolution can presage what's going to happen, right? The way that I would think about this is that the network's resilience to structural changes, such as the addition of new regulatory links, looks like it is adaptive and fosters evolvability because those networks that were impacted by structural changes that didn't work died out. Okay. Just like the reason why math fits nature so well is not because there's some uncanny goodness of fit between the mathematical equations that we have and the way that nature works. It's because through blood, sweat, and tears, mathematicians made mistakes, sent them to their colleagues found flaws, and that particular line of thought, that line of inquiry died out, crumpled up through frustration, maybe in the wee hours of the night, a mathematician toiling away at their yellow pad tears it up, says, no, this isn't working, and throws it out. That's what we have erasers for. So the selection here is hard selection against networks, and it's kind of a survivorship bias. If we're going to understand biology based on everything that survived and not understand everything that didn't survive, then we're missing the evolutionary story. So nevertheless, Albergante, that's where the BQS comes from. That BQS is when they study them as they might function in simulation, and then they looked at natural GRNs. They noted that stable GRNs that can persist are free from long feedback loops involving more than 3 genes, because more than 3 genes in a feedback loop leads to instability. Okay, we know something. So how many times did we get a feedback loop that didn't work, and then those organisms were left behind? All right. And then GRNs have a higher proportion of transcription factors that are not regulated by other transcription factors, reducing the risk of unstable loops. Well, that makes sense to me anyway, where if you have one transcription factor regulating another, rather than kind of more or less a one-to-one correspondence between a transcription factor per gene or 2 transcription factors per gene, but very few instances of transcription factors regulating other transcription factors, well, it would become unwieldy to be able to track the signal in that system, uh, and so too many cooks in the kitchen idea, right? So they looked at the data, empirical data. They found that real GRNs are significantly more stable than randomly generated networks of similar size and connectivity. Okay, that to me is evidence of the rejection of GRNs that didn't work, right? So that's evidence of adaptive evolution at play, yielding this rather than the BQS is allowing. See the difference? It's a big conceptual difference in evolution. Nevertheless, when we try to understand when BQSs go wrong, aberrant GRNs, okay, then the study found that GRN of a human leukemia cell line deviates from BQS predictions. Okay, leukemia is deadly. Sounds like selection to me. Evolutionary biology, understanding BQS provides insights into how GRNs evolved to maintain stability and adaptability. There it is again. No, BQS is evidence of the loss of those networks that did not have stability and adaptability. By definition, it's not in order to. This is a bugbear, and it's always been a bugbear of mine. I think I'm overdoing it on this point, but I hope that it helps you understand that when you go into this literature, which is a fascinating literature, you're going to you're going to get confused if you think that GRNs evolved to allow adaptability or to allow stability. The Albert Gante Blow and Newman paper is in eLife, Volume 3, published in 2014. Buffered qualitative stability explains the robustness and evolvability of transcriptional networks. Now, I could write to the journal and say I agree with everything that they said except for the semantic point, but everyone should know better by now, I think. All right, so I'm going to give you some life skills here. You can amaze and impress your friends and enemies by being able to read systems biology diagrams. Ooh, this will change the world. So if you see something in a systems biology diagram, like a square with an arrow pointing from the square to the circle, you read that as square increases the production or action or activity of circle. Right? That's what the arrow means. So it could mean that it activates it in a signaling pathway and a metabolic pathway. It tends to mean that A is produced from B, so it can get confusing. So make sure you know which kind of diagram you're looking at. With ligands and receptors, an arrow pointing from the ligand to a receptor indicates that the ligand binds the receptor and activates it. Okay. And then if we see this T-shaped diagram, this is used in systems biology quite effectively. If you see this T-shaped thing, then the square stops and blocks the production or activity of circle. So those two things alone, we could draw some pretty complex networks, such as this one on cell cycle progression, anti-apoptosis, and differentiation. So this is going to be a cancer one. You can read this fascinating UC San Diego thesis dissertation by Arya Venkat. And the reason why I chose this is they have some software published in 2017 that they highlighted in their dissertation called Path Insight. It's a novel tool for modeling biomolecular pathways. And there are a ton of pathway software, which if you take our bioinformatics course, you can learn 3 or 4 of them and get to work trying to understand pathways by looking at gene expression patterns or proteomic differences, combinations of genetic and genomic data, gene expression data. But the reason why I wanted to share this with you was because they, provided this comparison of their systems biological graphical system in Cytoscape, the Cytoscape being a particular kind of software. And so reading one systems biology diagram is not necessarily going to be an exact easy translation, but it's not that difficult. It's not like the difference between Sanskrit and English or something like that. So I wanted in this final part here to look at some key examples of gene regulatory network evolution. We're going to look at transcription factor duplication, regulatory network rewiring, look at some patterns of long-term evolution of GRNs and what's been found there. We're going to learn about and understand cis regulatory modules and their role in GRN evolution. And we're going to take a moment to reflect on Hox genes again. So with transcription factor duplication, you already have one transcription factor, and the binding site for that transcription factor itself might be north of or 5' to the gene, the protein coding gene. But the general pattern in the evolution of transcription factors after duplication of a gene is that you have a duplication of the transcription factor along with the gene. And then the transcription factors may become silenced, in which case that can become a pseudogene. The second copy can become a pseudogene, and it's free to evolve, and it accumulates non-synonymous substitutions at a faster rate than expected. but it's not due to selection. It's due to the fact that it's not active. It's not doing anything, and so um, you end up with it's free to evolve. It has degrees of freedom at the RNA level that it didn't have before. But if you just imagine the evolution of a transcription factor binding site, right? You can see this binding site duplicating on the same genome without gene duplication. So you can get two of these binding sites. And then that binding site itself would then be free to evolve, perhaps to respond to a repressor or an activator. So that's something that we can see and have seen. Um, and then if we look at when genes are in fact duplicated, they tend to lose their associated transcription factor binding sites, and they either become pseudogenes, as I mentioned, or they might take on a new function by being activated under a different milieu of signaling. If they have a local sequence, they're inserted into the genome where there's a local upstream sequence that has an activator or a repressor that already exists just by chance. And then, of course, through adaptive evolution at the nucleotide site, those transcription factor binding sites can evolve to be more responsive to the particular signal for repression or activation. I think that's really cool. And then polyzinc finger protein evolution is a very good example of all of this. Has been very well studied. They're a major source of new transcriptional repression activity in humans and other primates. They control the timing of stem cell progression, sexual development, immune control of viruses, and overall development. They're very important. Um, so they themselves have diversified across the genome as they have gone out to respond to the call, if we're going to think teleologically in evolution, to regulate other genes as those other gene families have also evolved and diversified, right? So it's like a master control switch here. And I used two with a tongue in cheek, as you probably know. So then imagine that you have a perfectly good gene regulatory network, and now it's going to be rewired. Now, we just talked about how that can be hazardous, right? But how can we get through the selection that would occur against individuals that had a faulty wiring of the GRN? Um, well, it turns out if you look at rewired regulatory networks of orthologous genes, that is, genes that are related to each other phylogenetically through the process of gene duplication, they actually contain a higher proportion of species-specific regulatory elements. And then there's the de novo evolution that occurs of transcription factor binding capabilities, like gains and losses of functions, evolution at the transcription binding site, like I mentioned, duplication or loss of copy number of transcription factor binding sites. Those things can all occur. So that is the extent of the complexity of regulatory network rewiring. It's all about the transcription factor binding sites, their number, whether they're present, their number, what their sequence is, what they respond to. But then it's also about the signaling molecules themselves, their shape. Do they bind to that particular nucleotide sequence or the double helix of that particular nucleotide sequence to open it up? So you can see differences in rewiring that involve silencing of a gene, upregulation of a gene, downregulation of a gene, or even alternative splicing, where you get different transcript factors off of the same set of exons because the different exons are stitched together in a different order. So it turns out that one of this reprogramming of a gene regulatory network, the rewiring, has been found to be important in fostering the development of life cycle differences in the biology and physiology and morphology of sea urchins. This, in particular, the study organism here is the green sea urchin. This is another PhD thesis. This one's by Naramani Dori. I hope I'm pronouncing that right. But the changes that occur in the sequences of the genome, look at the radical differences in the actual life cycle morphology here. Those changes that occur occur due to genetic regulatory network rewiring. Right, so that should be intellectually satisfying to see an example like that. Um, this is a related paper. Comparative developmental transcriptomics reveals rewiring of a highly conserved gene regulatory network during a major life history switch in the sea urchin genus Heliocidaris. And in this particular case, their study involved 3 sea urchin species um, that made their living in different ways. And looking at this for the development from unfertilized egg to early larvae, they could see that the life stage was the major descriptor of how the signaling in the network was actually structured. This is a principal component analysis, and I hope they're not suffering from the ubiquitous horseshoe effect on the second axis. They might be, but nevertheless. Um, and looking at this, then, here's a fantastic example from their study of the types of changes that occur across the different tissues. The darker the um, color here, the larger the shift. So there's some parts of the network that haven't changed at all with respect to lifestyle, where they make their living, how they feed. And there's some that have had major shifts in terms of their regulation and the signaling processes. So we're starting to get a handle on how to study this, right? So I really like this. Kind of looks like a blueprint, right? I I would be remiss if I didn't include, you know, a certain amount of detail on the evolution of methylation patterns because we're talking about gene regulation, and you can have the proper signaling as an activator, but if the DNA is methylated, you're not getting at it. So the changes in methylation patterns are known to be involved to the evolution of complex phenotypes by modulating those expression of genes in development, physiology, and adaptation. And the altering gene expression can change without changing the underlying DNA. So the evolution of methylation patterns on the genome itself would be a great way to get some variation that natural selection can work on if the methylation patterns, say, are heritable at the nucleotide level, which they are through CPG islands. So the addition of a methylation group to the DNA molecule, particularly at cytosine bases. So in other words, if there are certain changes that are necessary that can be controlled by methylation, then there's the potential, there's the opportunity for natural selection to, by happenstance, respond favorably to local increases in cytosine bases to enhance methylation. There have been significant changes in the human brain with respect to DNA methylation patterns, which is going to be fascinating to a lot of people because perhaps we can optimize our brain health a little bit by getting properly methylated. That's not a surprise to many people in my community, but the researchers in the study that motivated this part of the presentation found that differential methylation in regulatory regions contributed to the unique expression profiles of genes associated with brain development and function in the first place. These epigenetic modifications are believed to play a crucial role in cognitive and neurological differences between humans and other primates, and they are responsive to environmental factors. Okay. The methylation landscapes in the prefrontal cortex of human chimpanzees and rhesus macaques have been studied, and it was found that humans exhibit unique methylation patterns of regulatory genes involved in synaptic plasticity and neurodevelopment, which suggests these epigenetic changes contribute to human-specific brain functions. Another example where evolution, this would be considered microevolution within a lineage. Research on Tibetan populations demonstrated that DNA methylation changes at the EPAS1 gene are associated with high altitude adaptation. These epigenetic modifications facilitate efficient oxygen transport and utilization, offering a survival advantage in hypoxic conditions. So if you look at the Tibetans, they're living at high altitudes. They have unique adaptations to hypoxic conditions. And part of the adaptiveness of their genome arrangement includes DNA methylation patterns related to the hypoxia response especially EPAS1. Right? Flowering time in plants. The ubiquitous model plant Arabidopsis thaliana uh, have found that the FLC gene, which represses flowering, is dynamically regulated in response to environmental cues, so that the plants can synchronize their flowering. I just did it. So the plants thereby synchronize their flowering with environmental conditions, demonstrating the adaptive significance of methylation in plant evolution at the flowering locus C of the FLC gene regulatory region. Um, so, yeah, it's significant to the adaptation because flowering is very important, and the timing of flowering is very important. So the evolution of methylation patterns, complex phenotypes can be really profoundly influenced, modulating gene expression patterns in response to environmental and developmental cues. It's kind of a downer, right? Because if we could understand evolution just by inherited genetic variation, then we could all just do population genetics on populations of interest and understand the origins of the differences among subpopulations or subspecies or species and understand these patterns. But now we have to also look at epigenetic modifications for the origins of new traits. We look at long-term evolution here, complex gene regulatory networks can lead to an increase in the efficiency of generating beneficial mutations. What? Excuse me? Okay, here we go. Anton Krombach and Paulian Hogeweg, 2008, did a simulation. Oh, thank goodness. Okay, so we'll forgive them for the semantics. But the overview of the model here is that they have a simulated genome, and it is arranged by having specific regulatory networks that are defined. You get a gene duplication or deletion or other changes, and the relationships of what's turning on and turning off are done to alternate a particular pattern. A is on, then B, then A, then B. So they have a particular process in mind. I don't know what they're modeling there, but it's cyclic. And so then they come up with the generalization that once the network emerges, it tends to become resilient to large changes. So they start with random connections, and then it becomes resilient to changes once it firms up. Would be expected. Key genes regulating in a specific way can impact the efficiency of evolution. I love these claims. The rate at which the evolving networks achieve a certain functional threshold. So with the same mutational input, then a gene regulatory network is better at choosing via selection which mutations are going to go forward and which ones are not. That's how I would say that. Okay, so individual mutations can impact fitness more than changes in the network, most likely because large revolutionary changes are disallowed by selection. Aha! See, they said I was right. Large revolutionary changes are disallowed by selection. What they might be missing here, however, is that the mutations of interest also occurred in the networks that didn't survive. And so I want you to remember way back to the beginning, way back deep when we were talking about Darwin and Wallace and all the naturalists and the traveling and Galapagos and other places all around the world. And remember hopeful monsters. And I wrote an entire article about hopeful monsters. This is a hypothesis that suggests that perhaps their evolution transforms lineages via large leaps and macro mutations. The name associated with it is the German geneticist Richard Goldschmidt. He was the first scientist to use the term hopeful monster. He thought that small gradual changes could not bridge the hypothetical divide between microevolution and macroevolution. In his book, The Material Basis of Evolution, 1940, he wrote, the change from species to species is not change involving more and more additional atomistic changes, but a complete change of the primary pattern or reaction system. Whoa! Into a new one, which afterwards may again produce intraspecific variation by micromutation. Okay, so we have mutations that can change things, and we have networks that can change things through modular evolution. And these guys found in their simulation that individual mutations can impact fitness more than changes in the network because of that resilience. Well, I'm not sure they evolved modularity. They're looking at an individual network. So this would be like a single module. So we can't really assess that with their. Then we look at biology. There've been Richard Lenski has done amazing work uh, with long-term evolutionary experiences with E. coli in a lab over thousands of generations. Bacteria develop beneficial mutations in the same genes to make them, and therefore became more efficient. There I did it again, at adapting to new conditions, demonstrating increased evolvability. There it is again. I think I'm losing this argument. We'll come back to Linsky in a little bit. Cis regulatory modules are essential elements of DNA that regulate the activity of genes. Think of CRMs as the control panels or switches that determine when and where and how much a gene is turned on or turned off, like a dimmer switch. The CRMs are called cis regulatory modules. These are the ones that are on the same strand. Okay. They've been studied as a unique set of things for some reason, probably because they're important. They are crucial for the precise control of gene expression, which is necessary for the proper functioning of cells and development of an organism. This is how they work. They're located near the genes they control. They contain specific DNA sequences that can be recognized and bound by proteins called transcription factors. I've been calling them transcription factor binding sites. These transcription factors are like keys that fit into the lock-like sequences within the CRM. So you have enhancers, silencers, and promoters. We've already talked about these, but they've been studied, and you're going to run into it if you go into the literature. Their role in development and adaptation is very important to make sure that certain genes are only active in certain tissues. That's controlled by methylation patterns. And they also enable organisms to adapt to their environment by changing gene expression, enable. Organisms adapt to their environment by changing gene expression in response to external signals, survive, and those that don't do not. For instance, a CRM might activate a stress response gene when an organism is exposed to heat or cold. And if they don't activate that stress response gene, they're not likely to make it. Okay. So look at this alteration of cis regulatory modules that determine regulatory gene expression as a major mechanism of evolutionary change in GRN structure. Okay, so in this particular diagram, we're looking at domain 1 GRN and then domain 2 GRN. We have gene A that's turned on in the presence of the activation of 3 units, and gene B, the first 2 units are not activated, and you do not get the gene product. And then you can see they're just talking about mutations in the individual cis regulatory elements or the transcription factor binding sites and the consequences thereof, right? Based on mutations, even though you have the same environmental signaling on the right, because of mutations, you can still get the gene product. It overrules. Neat, huh? So you can see significant differences in how genes are expressed, contributing to the diversity of life forms. These changes can result in new traits or adaptations that can be beneficial for survival. Peter and Davidson looked at the evolution of gene regulatory networks that control embryonic development of the body plan, particularly looking at CRMs. And they have this diagram that shows the post-gastular axial patterning GRN in play geographically, overlaying and interacting with body part specific GRNs, overlaying and interacting with body part pattern formation GRN, overlaying and interacting with the sulfate specification GRN. That's how you should look at that, right? So then when you have these 4 modules, 1, 2, 3, and 4, all affecting these phenotypes, all of them their own semi-independent GRN with a regulatory control, then you get the output of the embryonic pattern formation overall. And then this particular paper looked at possible functional consequences of evolutionary changes in GRN. I thought this was interesting because it talked about gain of function, loss of function, changes at the sequence level, appearance of a new target site, across the board. You can get loss of function, quantitative output change, input gain loss within GRNs, and gain of function, cooperative redeployment to a new GRN. Cool. Nevertheless, we have appearance of a new target site, loss of an old target site, change in the site number, change in the site spacing, change in site arrangement, translocation of module to new gene, module deletion, new tethering function, and duplication, subfunctionalization. And their claim is that these cis-regulatory module evolutionary alterations can either do all four or are restricted. So changes in the site number changes the quantitative output only. See that? All right. So people have done a lot of thinking about the evolutionary consequences and the constraints on the different types of variation. As we saw, a good example of cis-regulatory modules and their evolution, the body plan development. Well, body plan development is what makes organisms of the same type different. What I mean by that is mammals and vertebrates are all different because of body plan development and many other differences, but that's a huge one when it comes to us recognizing them anyway. And how they live their life. A snake can't be a snake if it's got paddles, right? Transcription factor collectives look at cardiac cell fate determination, and they found cis-regulatory elements define cell lineages and reflect evolutionary history. So that's going to be probably related to methylation as well. And then in cichlid fishes, evolutionary changes of GRNs driven by alteration in cis-regulatory modules have been linked to the development of distinct phenotypic traits, underscoring the importance of regulatory changes in adaptive evolution. You know, it's arguable that this is a good example because the cichlid fishes hybridize almost as much as oak trees. But if you're interested in CRMs, and there's big literature on it, there's PLOS Computational Biology, on Body Plan, Journal of Biosciences, Deciphering the Complex Cis-Regulatory Landscape and Genome Biology, Evolution of Regulatory Networks Associated with Traits Under Selection in Cichlids. Let's go back to the Hox genes. We're getting near the end here. So you may recall that Hox genes themselves, if you've had my Bio-B, more organismal biology semester in biology, through those lectures, you will recall I fell in love with mollusks. And could I live my life over again as an evolutionary biologist that was studying diversification and speciation and specialization and adaptation? I would definitely study the mollusks. Hox genes play a very important role in terms of maintaining the bioplan. And as I mentioned earlier, their specific orientation on the genome and the order in which they appear in the genome, it turns out to be very important in terms of their proper regulation. Not a big surprise to you now, you're a GRN expert. And more than that, you're an evolution of GRN expert. I found it fascinating that, according to the fossil record, the molluscan vertebrate last common ancestor lost its encephalization relative to vertebrates. And this loss at the genomic level appears to be potentially, most likely, have been the result of loss of a lead controlling Hox gene at the 5' end of their Hox genes. And this, to me, my impression is that it allows there it is. As a result, the lineage ended up exploring other bioplants unrestricted by cephalization. So that's why we get weird things like snakes and slugs and in the same phylogenetic evolutionary clade as clams and winkles and mussels and scallops and squids and octopuses and limpets. It's just a huge diversity of types, seemingly unrelated, other than the fact that they lack a vertebrate and they could be a default garbage dump, but they're not. And for categorization, they're not. They play an important role in my understanding of the evolution of animals, right? And so each of those lineages specialized their bioplan by a combination of changes in the gene regulatory networks involving Hox genes and no doubt many other genes, and protein evolution. So to fully understand evolution, I guess the take-home point here would be that you can't really understand it just by point mutations. Beanbag genetics are not, it's not going to get us there. You have to understand systems biology and the evolution of systems biology and not be afraid of it, and go into the literature if you're interested in the organismal lineage, if you're interested in the evolution of intelligence in humans. You're going to run into gene regulatory networks, and I would encourage you to take the variation in gene regulatory networks as serious as you take other inheritable types of variation. And some references at the end. If you have questions about this lecture, you can send them to info@ipac-edu.org, info@ipak-edu.org. I'll be more than happy to answer any questions that I can. If you haven't signed up for this course, we have another 10 lectures. If you enjoyed this lecture, you can sign up at ipac-edu.org/registration. Upon signing up for Principles of Evolution, you will immediately receive 14 lectures, so you can catch up with the rest of the class. And I hope you guys have a great time.